Hi, everyone. Good morning. So let's continue discussing the nature of tumors in terms of how mutations give rise to tumors and why it is that in reality it takes way longer for mutations to accrue or build up to be able to generate something like an actual life-threatening tumor. It takes way longer for that process to happen than you might necessarily anticipate or expect. And we'll take a look at the role of things like DNA repair mechanisms in counteracting or detecting damage to DNA. So let's review some key concepts here. We've said that cancer is a failure of DNA integrity, leading to uncontrolled proliferation. And when I say lack of or a failure of DNA integrity, that means, of course, things like, well, mutations, right? Mutations that alter what the intended wild type sequence of our DNA is. You know, that sequence, that wild type DNA sequence, is the product of eons of evolution, finely tuning and tweaking things to get things to work right. When you think about all the proteins that our genome encodes for, right? We're talking about tens and thousands of different proteins that are coded for by our 20 to 25,000 different human genes. Each one of those genes, the DNA base pair sequence, is the product of evolution, right? To be able to tweak things based upon natural selection, leading to proteins having their shape being just right to work together in balance, right? So mutations that disrupt the wild type sequence of our genes is certainly a failure of DNA integrity, but also, right, our dependence upon different proteins in our cells to constantly monitor the status of our DNA and make sure that if there is any damage, that the cell, maybe does not go ahead and continue proliferating, but instead might need to stop, repair the damage, or if it cannot do so, the cell may need to undergo apoptosis, right? Again, that really kind of interesting way of thinking about it. An individual cell in our body from evolution has remarkable capability to adapt and adjust in order to stay alive, right? As conditions surrounding it get worse, get better, whatever, right? And so even though our individual cells come programmed to try to stay alive by upregulating certain pathways, shutting off other pathways, at a certain point, right, if things are too damaged, then those individual cells need to eliminate themselves, right? So in other words, the individual cell has to make that decision of undergoing apoptosis, right, to spare the rest of us, right? Our entire body continues surviving because that individual cell made the right choice to sort of give up and take itself out of the picture. We also discussed that self-renewing stem cells, right, persist in our tissues and give rise to replenishing our population of fully differentiated cells. Our fully differentiated cells that in relative terms do not stick around all that long, right? In some cases, you have fully differentiated cells that might stick around for a few weeks or so, but in other cases, you have fully differentiated cells that are only expected to last a few days. For example, those epithelial cells that line our GI tract, right? That's the example that we've used quite a bit. And we think about how those cells are shed all the time as a result of things like physical abrasion of food passing by them. But we also appreciated the fact that that's the intended way that these cells are supposed to function. Why? Because those cells that line our GI tract, boy, they are subjected to all kinds of weird stuff, right, that we're eating, all kinds of chemicals, compounds that might be mutagens that can cause mutations to happen and damage their DNA integrity, right? So that those differentiated cells are at very high risk of getting mutations. So, right, evolutionarily, we say that's okay because we only expect them to stick around for a few days. They're not going to stick around long enough for those mutations to really be a problem, right? Because by the time some of these cells get really bad mutations, well, whatever, they're eliminated from your body anyhow. And that's why last time we said, interestingly, evolution of us and how our bodies work is a constant balance, right? Of accepting the fact that, yeah, as we age, our ability to repopulate really good quality differentiated cells declines, right? Because our stem cells, right, they accrue wear and tear over decades of life. Right? And every time one of them gets a mutation, well, by definition, from that moment on, any fully differentiated cell that comes from that stem cell is carrying the baggage of that mutation as well. Right? But that's how we evolved. We accept the fact that we age, that our body breaks down, because we're balancing against the threat of cancer, right? So it's better for us just to get rid of differentiated cells and constantly depend upon stem cells to replenish them. Right? So here is a cartoon that I think it's a nice cartoon because it gets us thinking about, you have stem cells in your tissues that are self-renewing, right? That means the job of the stem cell here is to proliferate, right? And with every division, you're of course going to get two daughter cells. Here they're only showing one daughter cell coming out from it. Why? You say, where's the other daughter cell? Well, the other daughter cell actually maintains its stem cell property. So as this blue stem cell divides into two gray daughter cells, one of those gray daughter cells basically becomes the blue cell again, right? Because that is the stem cell that sticks around for the next set of divisions. Whereas that other gray cell, the one that's shown in the cartoon here, it begins to divide, and divide, right? And as it's dividing, it is differentiating further. Right? Decisions are being made about which sets of genes are switched on, which ones are left off, right? And those decisions then lead to a whole bunch of progenitor cells here. So these intermediate cells, all these gray ones, are what we call the transit amplifying or progenitor cells, right? Because ultimately, by the time they get to this last step here, and they divide one more time, and they sort of fully commit to becoming the differentiated cells in red here, right? From this initial first single division, after many, many rounds, you've made many, many differentiated cells from this one starting blue stem cell up here, right? Okay. So what that implies is that over time, right, mutations, because mutations happen more or less randomly, you have to account for the fact that as these cells are dividing over many, many years, that these cells don't just get a whole bunch of mutations at once. Right? Mutations will happen randomly at different times, and depending upon the particular cell type that you're talking about. Remember the normal origin of where this cancer came from based upon the different types of cells in your body. Remember, you have something like 220 different cell types that make up the human body, right? And then those 220 cell types, you have trillions and trillions of them, right? So each cell type started off or follows particular pathways of gene programs, right? Different sets of genes that are more important for a certain cell type and maybe less relevant for a different cell type. So it's not surprising then that the progression of cancer might happen at very different typical rates, depending upon what the original starting tissue is. So let's look at this. Right? So this figure here depicts sort of visually a series of changes that takes place over time, starting from normal tissue. So let's say that this is, normal tissue here. And the over time, there's going to be one cell in here that gets a hit, let's say. So this cell is marked as red here, right? So this is the initiation of transformation, right? And then the next step would be maybe hyperplasia. So then this cell divides rapidly, so the population of cells becomes more and more sort of represented by this single cell because the cell is dividing a lot more than its neighbors, right? So we say this is sort of precancerous now. At this point, this is just hyperplasia, meaning the overall morphology of the tissue, more or less is still the same. The individual morphology of the cells, is only beginning to change, right? But you can see structurally, right, you can still tell that there is some maintenance of the original tissue organization here. But of course, as we've learned, as mutations build up, right, and these cells become more and more cancerous, right, then you really begin to see loss of proper morphology. Right, then control over proliferation is just sort of out the window now. And then you have really now a lot of changes in morphology, right, eventually leading to what we call carcinoma in situ, right? So IS means in situ. That means this is still taking place at the original site where this first transformed cell was originally found, right? And as long as this highly dysplastic tissue is still contained in that original site, right, it is becoming carcinoma, but it is still contained, right, in situ, meaning where it first started. Once those cells build up enough mutations that they learn how to break free of physical limitations, for example, the basement membrane, if we're talking about epithelial cells, and they begin to then spread to surrounding tissues, that then becomes full-blown cancer, right? So you can see the cells here are now busting through this, imaginary basement membrane here. So these steps, though, remember how we said that in reality, in an ideal situation, right, if you had one cell and it was just left to divide, right, unchecked, after something like whatever it was, 40 rounds of replication, the number of those cells would be exponentially large, so you'd actually have a fatal tumor size, right? And we said that, well, that's kind of weird because if a cell, let's say, begins to lose control of proliferating and it only proliferates or divides once a day, then doesn't that mean like in 40 days you'd have a lethal tumor, right? And we said, no, that's not true, because along the way, what's happening to most of the daughter cells? Along the way, most of the daughter cells are doing the right thing, meaning they're detecting that there's something wrong with them that they cannot fix, and therefore they undergo apoptosis, right? So that's why we said that in reality, you might have one starting cell, and after 12 rounds of replication, you would expect to already have thousands and thousands of them, but in reality, you might only have 5, right? Or something like that, right? The more generations you have, right, the more time it takes. And the more time it takes, of course, is more opportunities for other random mutations to start happening and start piling up as part of the baggage that gets passed on. So let's look at different examples of cancers here. So colon, head, neck, cervical cancer, lung cancers, breast cancer, prostate cancer. I think this is a really nice figure because this really helps you appreciate the fact that because these are different normal starting tissues with different types of pathways that are important in them, the progression of cancer is quite different. For example, in colon cancer, typically from the time when you have a single cell that is transforming in there to the point where it actually starts forming a tumor, so in this case, an adenoma. So let's say this is a gland cell in your colon, right? Lining of your colon, maybe the crypts or something like that. It begins to transform. It can take about 5 to 20 years, right, of many generations of that cell dividing before you even start forming something like a small polyp, right? A small benign lesion in there. At that point, from the point where it's becoming precancerous, the adenoma, it might be another 5 to 15 years before you actually reach the point where it's full-blown metastatic cancer, right? And it is spreading. So that means from the initiation of that one transforming cell to the time where you actually have malignancy, it could be something like what, 35 plus years, right? Look at something like, you think that's a long time, right? Think about something like cervical cancer, right? From the time that you have a transformation taking place, it might be 9 to 13 years before you get to what we would call, cervical intraepithelial neoplasia, sort of phase 3 of it, right? And then that moves on to carcinoma in situ, and another 10 to 20 years before that makes its way to becoming cancer. Some cancers are very different based upon things like the exposure to mutagens that cause the cancer in the first place, right? Lung cancer, for example, right? There's no real well-defined kind of time frame for that because it has to do with how much the person smokes. But you can imagine over the course of 20 to 40 pack years, right? That means smoking a pack of cigarettes a day. That's how long it takes for you to eventually get the development of full-blown cancers. Now, that varies from person to person, of course, right? Some people hardly smoke at all and they develop lung cancer at a shockingly early age. Some people, they smoke like chimneys every single day and never develop cancer, right? Again, because of things like the randomness of mutations, right? So again, hopefully this allows you to appreciate that in reality, the formation of a lethal tumor, right, is not something that happens overnight, right? It happens over many years. But, however, I think all of us, right, have experienced somebody that we know who was diagnosed with cancer, right? Family member, friend, or what have you. And it often feels like from the time you first hear about, oh, so-and-so is diagnosed with cancer, to the point where they end up, unfortunately or sadly passing away from that cancer, it seems like a really short time, right? When I was, after graduating college, I was living in Washington, D.C., and I remember one day my mom, who's in California, she called me and she told me that my uncle Ken was diagnosed with pancreatic cancer, right? And at that point, I had seen my uncle Ken, pretty recently, within the last few months. But now I was all the way on the East Coast because he's in California also. And I said, oh, that's terrible, right? I wanted to kind of, I felt bad and I wanted my mom to update me, how is Uncle Ken doing? And then it was like 2 months later, she called and said, oh, he's undergoing chemo for his pancreatic cancer. I'm like, oh, okay, and then, 6 months later, he died, right? So to me, it was a shock because within like what felt like such a short time, we found out that he had cancer and then he was gone, right? So to me, I thought, wow, when cancer starts, right, it progresses really quickly. When in reality, the truth is that last part that you actually know when somebody has cancer because they are symptomatic and being treated, that is already right at the end of this particular slide here, right, that I'm showing you in this PowerPoint. And you had no clue about the years and years leading up to it, right? So cancer progression takes many different pathways. So here's an example from the book where we're looking at one kind of hypothetical situation where you have a patient who develops colon cancer. And these are sort of the different stages again, or not stages, different phases that the cells are going through as part of turning into cancer. So you start off with normal epithelium, and then, one day, unfortunately, one of the cells takes a hit. Maybe that mutation just happens to be a mutation that disrupts the normal functioning of the APC protein, right? So the APC gene gets a hit in it, and as a result, the APC protein is no longer the right shape, or for some reason, the mutation causes a loss of APC expression. So either way, the cell loses functional APC. And remember, APC is important because APC helps control beta-catenin signaling. Remember, beta-catenin is one of these examples of a transcription factor that's very important for controlling genes that push the cell forward through the cell cycle. Normally, our cells produce a lot of beta-catenin, but what? That beta-catenin is constantly being turned over and degraded, right? Because it associates with complexes that have APC in it, resulting in the beta-catenin being labeled for degradation. So if you don't have APC, then that beta-catenin kind of escapes from being degraded, and now you've lost control over beta-catenin signaling. So let's say this is the first unfortunate by chance hit. So that results in hyperplasia, right? So this cell begins to divide more than it should. Right? But again, right, as it's dividing rapidly, most of the time, that population of daughter cells, right, those daughter cells are hopefully, right, detecting that something is wrong here, and they undergo apoptosis, right? But you might have a few daughter cells that for some reason don't do the right thing, and they keep going and going and going. Then potentially sometime later, those cells get a second hit. In this case, there's some hit that results in DNA hypomethylation. Right, so as a result, methylated CpG sites around the genome begin to lose the methyl group. Right? And remember, methylated DNA favors heterochromatin formation, right? The compaction of chromatin to be transcriptionally repressed. Right? So if you remove methylated DNA signals, then you favor heterochromatin unraveling and becoming more euchromatin-like, right? Which favors transcription to happen. So methylation and changes in methylation are a very important part of the progression to cancer. Overall, as cells transform, we see on a global sense throughout the genome evidence of hypomethylation taking place. Right? Most of this hypomethylation takes place in repeat regions of our genome, so non-coding regions that are oftentimes these repeats that maybe have their ancestry in viral genomes, things like that. Right? Those are usually tend to be sites of methylation. So those tend to open up when they're hypomethylated, right? The actual way that contributes to the progression of cancer is not well understood, but most likely it has to do with the fact that these repeats, should be normally methylated so that the structure of the chromatin around these repeats is condensed, so that the overall things like the way that the chromosome can loop around and have things like enhancers talking to promoters of genes somewhere else, right, take into account the fact that these repeat regions tend to be methylated and condensed. So maybe when you unravel these regions, that messes up regulation of other genes that depend upon proper looping of the chromatin. Right? What is interesting is that I said globally, you see hypomethylation take place during the progression of cancer. But if you look specifically at the CpG islands that are in the promoter regions of genes, right? So those are those regions where you have many CpGs in repeats, right? 20 or 100 CpGs. What you see is hypermethylation taking place. So that's kind of weird, right? That means there's a tendency at CpG islands and promoters for hypermethylation to take place, shutting down the expression of certain genes, right? Whereas overall, globally, the cell experiences hypomethylation. So again, the two must work in some ways that are not always kind of set in stone, right? So it could be that hypomethylation of the repeat regions leads to loss of proper regulation of other genes that are nearby, and hypermethylation of promoters of genes are shutting certain genes down, right? And you might say, well, wait a minute. Yes, we already looked at how hypermethylation of promoters of tumor suppressor genes is a big part of the progression to cancer, right? By shutting down tumor suppressor gene expression like retinoblastoma or P53 or P21 or P10, right? That obviously is a way that you're releasing the brakes on oncogenic pathways. But you can argue, but aren't those oncogenes themselves, right? Aren't their promoters also being hypermethylated? Yeah, right? So it can be very well, it could be the case that you have some cells where certain promoters of oncogenes are hypermethylated, and as a result, those proto-oncogenes are turned off, right? But again, you don't see that. Why? Because if you have a cell that has all these proto-oncogenes turned off, what happens to the cell? It does nothing, right? It no longer proliferates, and then it just eventually just disappears, right? So you tend to see those cells where hypermethylation is taking place at tumor suppressor genes, because those are the ones where if you release the brakes, right, then the cell moves forward, right? And you are sort of biased towards seeing those, right, as the example of hypermethylation. Okay, anyhow. So then, after a certain amount of time, right, it starts developing into a benign lesion like an adenoma, right? But then you have a third mutation, a third hit that, for example, activates KRAS, right? So now you've lost control over KRAS, and now KRAS is functioning. Then you have another hit, loss of some tumor suppressor gene, maybe at 18q, right? And then finally, loss of p53, right? And once you've reached that point and you've lost p53, then it's really like things are beginning to move ahead rapidly, right? And you end up with carcinoma, and very quickly after that, you get invasion and metastases. So let's think about this possible pathway that this colon cancer in this particular patient may have gone through. So the first hit is loss of APC, and we see that in 80% of colon cancer biopsies, right? That there's evidence that APC was lost at some point along the way. And so by kind of almost reassembling retroactively a timeline here, right, researchers have determined that this most likely happens as one of the first random mutations in the majority of colon cancers, right? So mutations in beta-catenin itself, right, that might contribute to loss of beta-catenin are also very prevalent, 15%. So let's say that in this case, right, this cell took a hit in APC. The next step could be any one of these over here, something that disrupts the RAS pathway, right? So it could be, for example, mutations that affect the expression or activity of KRAS itself. So 35% of colorectal cancer biopsies show evidence of KRAS, right? Similarly, right, 32% of them show some mutations that disrupt the function of PI3 kinase, and other ones such as BRAF that are part of the MAPK, the RAS MAPK signaling pathway, right? The third mutation might be loss of one of these tumor suppressor genes that are on chromosome 18, so TGF-beta or SMAD. Remember, we said SMAD is a very important protein that controls MYC function, right? So you can see in colorectal cases, cancer cases, there is high prevalence of one of those being lost, right? And then finally, they predict that the majority of colorectal cancers, right, then get a hit in something like p53, right, that finally then leads to carcinoma. So this is kind of a wandering, meandering path, right? But hopefully this helps or impress upon you two things. One is that in colon cells, like colon gland cells, for example, the intracellular pathways are such that this series of events is one possible way that it can lead from being a normal cell to a cancer cell. But at the same time, the actual mutations that sort of make it progress along this way can be varied, right? So many cancers have that type of variation. There's many meandering different routes to get you from point A to point B. Certain cancers, though, right, are more straightforward, like the example of pancreatic cancer. So pancreatic cancer is one of those, more unusual cancers where things progress very quickly due to the fact that there's a pretty well-established pathway that these pancreatic cells go through in terms of the types of mutations. Right? So this is your progression from a normal pancreatic cell to invasive carcinoma. And you can see that KRAS activation takes place in over 90% of these cells, followed by, almost all of them lose P16 function, right? And then the majority of them, 75%, then lose P53, and then most of them then lose SMAD4, right? So again, right, this is a series of random mutations that if they follow by chance this particular set of hits, right, this is how you end up selecting for cells becoming cancer. Whereas the majority of cells along the way that are going down this pathway, they fall by the wayside, right? Either because they undergo apoptosis or the random mutations they get, right, don't actually encourage this particular progression to happen. Right? This is a nice way of visualizing the time component of this. So let's say here we have an array of normal, healthy, differentiated cells in gray. So this is all the cells that are part of, some tissue in your body. And let's say one day, one of them gets this hit, right? So this would be the initiating mutation that kind of starts it transforming. So that hit most likely is a hit that somehow encourages proliferation, right? So maybe a hit in some sort of proto-oncogene pathway. As it begins to divide, it divides a bit faster than its neighboring cells, right? Because now it has a growth advantage in the sense that one of these proto-oncogene pathways has been disrupted. But as it divides, right, most of the daughter cells along the way do the right thing and detect that there's something wrong, right? And they undergo apoptosis. That's why by the time this first transforming cell has a chance to divide enough over many, 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 many generations before it reaches, let's say, 1 million of these cells, right? That's taken something like 10 years, right, or more to happen. Because up until that point, it's only had this one mutation, right? And most of the cells along the way, right, have disappeared because of apoptosis. But after 10 years, you have, let's say, 1 million of these cells. So what does that mean? That means now there's a much greater chance that one of these cells that is already carrying that red mutation gets a second mutation randomly, right? And that second mutation might do something like further increase the proliferation rate, or this might be a mutation that is now releasing the brakes, right? For example, a mutation in a tumor suppressor gene. So as a result, with that increased proliferation rate, right, the amount of time it takes in this second clonal expansion is shortened. Right? So instead of 10 years, it takes maybe just another 5 years for this cell or this new version of this cell or this subpopulation to multiply that has with it, each one of these has the red mutation and the green mutation to start with. But now what's happening? Now you have so many of these cells that you have different random ones of these cells getting the third hit, right? Whether it's the yellow hit, the dark blue hit, the turquoise hit, pink hit, whatever. And each one of these now, these additional hits, not only are increasing the rate of proliferation, they're also increasing the rate of mutations, right? Why? Because now you're getting hits that are disrupting the ability of cells to properly repair DNA, right? And put an end or stop to these cells dividing. So as a result, from this point on, things might happen very quickly, right? And importantly, the resulting tumor mass is made up of these different subpopulations of cells with different potential phenotypes because they all have different mutations that have messed up different pathways in them. Although all of them originally started, 15, 20 years ago from this one cell that got that first initiating mutation. So a resulting tumor mass then, by the time a patient presents in the clinic with a life-threatening sized tumor, right, that tumor is a mess, right? Because it's no longer a homogeneous population of cells. It is a hodgepodge of different phenotypes. So that's what we see here. So here we have a cross-section of a pretty advanced tumor. So everything you see in here is practically cancer cells, right? But what you notice is that these cells are very different from each other. The little dotted lines here represent different sort of subpopulations of cells. And you can see some of them phenotypically, they tend to form very dense masses of cells, like this blue one on the left. Others, right, form much more loose, messy pockets of cells, maybe because they have a lot more fat or lipid deposits in them. That's what these white spaces are, right? Still others, like these green ones or the red ones here, in particular the red ones, looks like they are actually forming different structures, right? So in this case, it looks like these are actually forming new vessels of some kind, right? Because this is a cross section, and it looks like they're forming the walls of some sort of new type of duct or vessel. Right? And that's all because the phenotypes in these cells are getting progressively more and more further away from each other in terms of their genetic makeup. And each one of them is now experiencing certain genes being turned on that should have been left off, and certain genes that are being turned off that should have been left on. And in extreme cases, that level of random disruption can actually turn on complete genetic pathways that are coded for by the genes, right, that these cells carry with them, because all of these cells in the picture here, they all started with the same genome, right? So all of them started with the same set of genes. But now some of them, like the ones in the red here, right, may be turning on pathways that they have no business turning on. For example, pathways that form ducts, right, or vessels. In really extreme cases, right, you can actually have them form tissues that seem really, really out of place in the tumor. So there are certain classes of tumors called teratomas, right? And just like as the name implies, right, they are terrible tumors, right? That's why they were named terrible, because they are horrible in their phenotype. So these are tumors that oftentimes have within them, they would be this big messy ball, right, of tumor cells, but oftentimes they contain things like hair, they contain things like teeth, believe it or not, right? Or even the formation of early eyes, all right, in these tumors, right? When I was a little kid growing up in the 80s, we had these things, these mad ball toys, right? Where these sort of round rubber balls that had all these horrible, ugly kind of like Frankenstein, pig-nosed kind of like faces on them. I don't know why they were so popular. I as a little boy, I thought they were cool because they're really gross, right? You could scare the girls with them in school when you took them out of your backpack. But so teratomas, right? I'm not going to show you a picture of one. I don't want to necessarily encourage you to go online and look for pictures, because hey, I know some of you will do that. But it's the type of thing where if you see one, you're like, okay, I shouldn't have seen that, because now I can't get it out of my mind, right? Because literally in some extreme cases, you have things like surprisingly well-formed other body parts as part of the tumor. All right. So let's talk about how our cells have to deal with, right, this constant threat of mutations taking place. So mutations that take place that can lead to the development of cancer, right, might happen from things like mistakes during DNA replication. When the cell enters S phase as part of totally normal cell cycle progression, right? Let's say a stem cell, right? Stem cell, that's whole job is to proliferate, right? As it goes into S phase, it has to successfully make a perfect copy of the genome that ultimately in mitosis is going to be divided into the two daughter cells. Mistakes or damage can also happen through endogenous biochemical processes. Just the fact that our cells are alive, right, and they're engaged in metabolic processes. Many of the waste products of metabolic processes are damaging to biological molecules, like to our DNA or to proteins, right? And thirdly, of course, when we think about, what are things that cause mutations? I think our sort of knee-jerk reaction, and if you were to ask somebody on the street, average person, hey, what are factors that cause mutations? They would say, oh yeah, exogenous factors. Well, a typical person off the street wouldn't use exogenous, but they would say like, yeah, things that give you cancer, right? Like chemicals or UV radiation, right? But let's think about all three of these possible mechanisms. So let's start with mistakes that happen during DNA replication. And then in the next lecture, we'll look at points 2 and 3. Okay. So as our cell is going through S phase and it's replicating our DNA, I think all of you appreciate what a tremendous undertaking that is, right, to copy all 3.2 billion base pairs of our genome and to do it with accuracy, right? Because as we said, if one single DNA base pair is altered, depending upon where that DNA base pair is, it can have very bad consequences, right? Because that original wild type DNA sequence, it is what it is through millions of years of evolution and getting it just right to be what it's intended to be, right? So in the process of copying DNA, you're bound to make mistakes. I used that analogy in another lecture about how when we think about making a copy, right, of something like a photograph in a photocopy machine, we all accept the fact that even if it's the original photo and you're just looking at the first generation of photocopy, color photocopy that comes from it, right, there's already going to be some information loss, right? The photocopy is not going to be as nice as the original copy, right? So think about when you replicate your DNA, the same principle applies. There's going to be mistakes along the way, and it's up to our cells to make sure that those mistakes are caught and fixed, right, before the cell is allowed then to actually distribute that copied genome into one of the daughter cells. So that proofreading of DNA starts with the DNA polymerase itself. So here is the complex of different proteins that makes up DNA polymerase, right? In this case, it's reading a template strand. The green strand is the original starting template DNA. And as it moves on in a 3' to 5' direction, because remember, DNA polymerase can only go in that direction, it is assembling, right? The new copy strand, the blue one here, it's being made in its 5' to 3' direction. Now recall that the way the polymerase assembles the strand is there are free-floating nucleotides surrounding this, right? And if the correct nucleotide happens to float into the right position, it'll be stabilized by the fact that the template strand has the complementary base there, right? And that stabilization provides enough time for the polymerase protein complex to go ahead and actually make the phosphodiester bond that links that base as the new end of that growing strand, right? Which direction am I going in? I guess I'm going in this direction here. But if you think about it, right, if it's all has to do with these nucleotides floating around and they're all just kind of like, randomly getting in there, some of them get in there, but the wrong ones, they fall out, right? It's quite possible that you have the wrong base, right, that gets in there and the polymerase ends up adding that as the next strand. So that's, or as the growing strand, right? So that's what this little thing is over here, right? Oh, so by the way, I don't know why the book represents the polymerase complex in this particular shape, because this is not a shape that I typically have seen the polymerase represented in, but it's meant to highlight the fact that it's many proteins working together. I think it's kind of a weird shape, right? In fact, when I looked at it, this is one of those examples where the first time I saw this, I kind of stared at it for a long time because it was like reminding me of something, right? And so I realized, oh yeah, this looks like some really trippy, like Beatles' Yellow Submarine kind of thing, right? I always have these like flashbacks to like, the 70s or whatever. Okay, but anyhow, so DNA polymerase has proofreading ability, meaning as it moves forward, it can actually back up, and that's what it does. It moves forward, it backs up, and it proofreads, right, the bases that it just added. And so in this case, if it backs up, right, it can detect that that base that was added last is incorrect, right? And so, so it's kind of funny, the little proteins, these little lines here, I'm not sure exactly what's going on. They're like jiggling or something like that. No, I think actually what it's showing is that it's signaling for other proteins to come and remove that misincorporated base. And then the submarine can then proceed to move forward again, right? So now it's jiggling in this direction, right? So that's proofreading that is just a function of the DNA polymerase itself, right? What if the proofreading of the DNA polymerase misses something, right? And then you have a misincorporated base that is now part of the template strand. Now it is up to different mismatch repair or MMR enzymes to come and recognize, oops, the DNA polymerase made a mistake and then it didn't catch the mistake. So now these other proteins have to catch the mistake and fix it. Right? So here you can see there's an error in the newly made strand, and as a result, it's not the right Watson and Crick base pairing across the two strands, right? So the alpha helical shape in that region is not the normal alpha helix, right? You can, here in the cartoon, it's represented as this funny little bulge, right? So you have proteins like these mut-S and mut-L proteins. These were originally identified in E. coli, by the way, but of course our bodies have homologs to them. They are constantly associating with the newly formed DNA and sort of checking things out, right? They ride along the double helix looking for these distortions in the shape of the double helix. And when they encounter one, you can see they're scanning, they detect a break in the strand, or sorry, they detect that there's a disruption in the strand, right? And they will go ahead and initiate the removal of that section of the DNA so that polymerase can come back and fill in the strand and hopefully get it right. So how do these proteins actually detect something like a bulge in the DNA helix, right? Because it makes it seem as though somehow, oh, they're pretty smart, right? They know what they're looking for, right? But the reality is, right, they're just proteins. How do proteins typically detect things? They detect things because proteins are based upon the right three-dimensional shape, right? And if you change the shape of a protein, you provide the ability to activate certain things, right? Such as changing the shape of a protein to reveal different three-dimensional surfaces on the protein that can then attract other proteins. And so here is a nice sort of crystal structure of those mut-S proteins associating with a kink or a bulge in the DNA double helix. So here in this double helix, you can see there was the misincorporation. So there's an improper base here. So as a result, it does not base pair properly across the DNA ladder. And as a result, even though leading up to this spot, the DNA double helix looks pretty good, when you get to the spot here, it's distorted, right? So there's like little bend in the DNA here. So here comes these mut-S proteins. So these are different domains of these mut-S proteins. These are not the full proteins themselves, because the full proteins themselves would be really big in this picture. And you would not actually be able to see the part that's going around the DNA. So now we're just looking at the peptide sequences that are surrounding the DNA here. These mut-S proteins are basically riding along the alpha helix, sort of like, you know, like a roller coaster. They're going around and around and around because they're the right shape to match up with the double helix, but they get to this bend here, right? And they get stuck. And when they get stuck, the bend causes the shape of the proteins to change, right? And once the shapes of the proteins change, then suddenly other proteins that are the right shape to bind to those proteins get brought in. And those are proteins that are then involved in the removal of this segment of the DNA and also the recruitment of other DNA polymerases to come and repair or fill in, right, the missing bases. So let's think about this then. This is the last slide for today. DNA repair, right, during replication, it better be very efficient, right? Because hey, if you're talking about accurately copying 3.2 billion base pairs of DNA, right, and you can't really afford, you know, to kind of screw up in critical places, then you better believe that this process is pretty efficient. So let's look at some of these rates of errors. So the DNA polymerase itself, so the submarine, as it's going along and making the new copy strand, it makes an error in about 1 in every 10 to the 5 bases, right? 10 to the 5 is what? Every 10,000 bases, it makes a goof, right? And it puts the wrong one. But it's okay because it has that ability to back up and do proofreading. So the proofreading, though, you know, it's a little sloppier, right? Because the 3 to 5 prime proofreading misses about 1 in every 1,000 mistakes. All right, so that means the submarine moves on and leaves the mistake. But then you have the MMR enzymes. So the MMR enzymes that come cruising along the alpha helix, right? They detect these kind of distorted DNA shapes. You know, they make a mistake in they miss maybe 1 in every 100 bases that are actually wrong. Right? So this actually starts then from the DNA polymerase itself being very accurate, And then progressively, the different layers of error checking being a little bit more lenient and a little bit sloppy. So the overall miss rate then, if you take all of this into consideration, is about 1 in 10 to the 9, or 1 in every billion bases, might actually be wrong, right? And you think, well, our whole genome is 3.2 billion base pairs, right? So that's pretty good. So that means what? Every time our genome is replicated by one of our cells, we get what? We have about 3 new mutations per cell replication, right? So that's not bad, right? You think about like the photocopy of the photo in the photocopy machine that I described to you, right? When you look at that, you can really tell, yeah, this is not as good as the original photo. It almost like that would be like a higher error rate than just 3 bases per entire genome, right? But I want you to consider this. That is in just one cell undergoing replication, right? Think about how many billions of cells in your body are replicating right now, all of your stem cells. So when you actually extrapolate this, that means if we have about 37 trillion cells in our body, that means your genome every day in your body is dealing with thousands of new mutations just as a product of things like your stem cells dividing, right? And so that is the baggage that you are accruing every single day, just by the fact that you're alive and that your cells divide, right? So now you have a new appreciation for how important it is that just by cell division, right, mutations happen. And that's why we said before, our bodies are designed to last us 35 to 40 years, right? Mother Nature designed us to be able to tolerate this level of mutation over 3 or 4 decades of life. But now that we live to 80, 90, 100, right, this buildup of mutations, especially in our stem cells, right, has a big, profound impact on the development of cancer later in our lives. All right, let's go ahead and stop there.